Hi guys, welcome. I'm just waiting for the others to join. Hi. Welcome, welcome, welcome. Welcome, guys. What's on the list today? Oh, I don't know. There's like a whole load of things. I've kind of been going back and forth, back and forth. I've just seen paper 2 and had a quick go through it, which was yeah, it was interesting. Oh, bless you. It's hard. It's hard to know because you have a vague memory of what you did, but actually there'll be things that you think you've written that you won't have written. There'll be things you've forgotten. It's really hard. But I did go through. So in the time, I've had very little time. So my brain's kind of all over the place in what I'm going to be doing. But I have gone through the paper. I've had a quick go through. I've not done it like you guys shouldn't because I did not have enough time, but I tried. And there's a few tricky ones in there. Even I'm thinking, because I was going so quick, I'm not sure what they're looking for in some of those questions, but it was good for me to see. I think the questions and the way they've asked them are quite confusing. So I can see why a lot of you have come out going, I'm not sure what they were wanting because I'm still a bit like, oh, I think I know. I think I've answered it in the right way, but I won't know for sure. But a lot of you kept saying to me last night and kept going, oh, practicals, practicals, practicals. So I did a bit of work for you. My daughter's not happy with me. My youngest has been like, Mummy, why are you not with us? So I'm going to have to be really nice to her tomorrow. Because this is not normally what I do, but I knew because tomorrow is the big day and after tomorrow I'm going to sleep and enjoy the rest. I wanted to do a bit of prep for you guys in that sense. So the first thing I'm going to say is I'm going to show you, I went through the specification again. It's been something I've been meaning to do. And what I wanted to look at is show you where the different packs specifically come up in which topic area. So as you go through the specification, you'll see a bit that says PAG 1 or PAG 4 or PAG 5 and whatnot. So I want to just go through and show you where they could ask you questions. Now, the one thing I did notice is obviously they've already given you a picture of the cells, and they've given you a picture which was showing you the mitosis stage. So in that case, do I think they're going to do that? I'm not sure, but I still wanted to put it in here so you could see where in the specification it links to PAG 1. So when it comes to microscopes, and I feel like they've done a lot of that because that first one on stem cells was quite clearly very much microscope heavy. But you've got your cell structure, cell diversity, exchange surfaces, transport plants. So what I've done is I've gone through every single one. So this is PAG 1. And obviously you guys can go back and read this in your own time. I'm not going to spend too much time on each individual part, but I wanted to show you this. This is PAG 2, so dissection. Now, they haven't done anything on dissection side of things, so that could potentially come up. And obviously you had your fish and your insects in paper 1, so do I feel like they could come up again? I'm not sure, to be honest. It's one of those things that I definitely feel like you're going to get something that you're going to have to draw. And so doing a drawing of whatever they decide to give you is where I think they could go. You better enjoy your summer too. I have been very hard working. Oh, does it keep glitching? That's not good. I hope it sorts itself out. But yeah, going through this, it's definitely one I would say to just make sure that you are confident if they ask you anything. Now, I want to highlight in here because I don't know why, but I've got such a strong feeling that this is coming up. So transport in plants with dissection. So the key thing to note, the dissection of stems, both longitudinal and transverse, and their examination demonstrating position and structure. But what I also want to bring in is, if I can find it now, I think it was in the one above. Yeah, if I go to the one above with PAG 1, the examination and drawing of stained sections of plant tissue, and the distribution of the xylem and phloem, I feel like that could potentially come in again, because they haven't done the translocation yet. And I do have a strong feeling translocation's coming through, because from all the things I've seen, they haven't spoke about biological molecules much. They mentioned it in that last question at the end, but I don't feel like it was enough. I'm planning on going with this live. I will probably go no longer than 2 hours. I'll see how I get on. I'll start answering questions. What would you draw? Whatever they give you as a picture. So when they're asking you to do a biological drawing, they're not expecting you to do it from memory. What they're expecting you to do is to look at the picture that's in front of you and draw what you see in proportion. So it could be, for example, that they give you a tissue showing you the phloem and showing you a companion cell. So you'd be expecting the phloem sieve tube with a companion cell alongside it if they were going to give you a picture of that. I don't know what they're going to give you. So it's that whole thing of I can make educated guesses, but I don't know. It just depends. Do we get a box? Yes, you will. And when you get that box to draw your diagrams into, just like with a graph, and they haven't given you a graph yet, so I do feel like you're going to get a graph coming this time round. Interestingly, on the question, which was about variation, they gave you continuous and discontinuous variation in that question. And so therefore, I still, again, have a strong feeling, and I could be wrong here, guys, but I have a feeling that a histogram's coming. You should do a title, you should have a scale bar, you should have annotations to your diagrams. One of the things that comes through quite a lot, by the way, in the examiner's report, if you are unsure, like I am a very visual person, I love doing diagrams because in my head, I can pull things forward with a diagram more than I can with writing things out. So therefore, what I would say to you is, you're not going to be penalized for drawing diagrams, okay? But why you don't get the marking points when you do a diagram is that that diagram should be annotated. You need to annotate, not just draw the diagram. So you need to be annotating whatever it is that you're trying to show if you do it in a diagram part. So I feel like you're going to be drawing a graph. I do feel like you're going to do analysis of some data. That's just a given. That's like guaranteed you're going to do that. But it really just depends on what they give you. Yes, you could use, Grace, a diagram for zero dilutions. Absolutely. Just make sure it's very clear. How would you be assessed on your histogram? So again, it will be your axes, it will be your scales, it will be your size and width of your bars, making sure your bars are touching and that the widths are correct based on the data that you've been given, and that your points are plotted correctly. There'll be a lot of valuation. I do feel like an evaluation is coming in. I felt the same thing when I was going through the paper just before this session. I was thinking, I haven't seen a question that says evaluate, or there was a conclusion, but there wasn't an evaluate on the information that's been given. There was the question in paper 1, which was about a null hypothesis and how you would then conclude on that front, but I don't feel like there's been one where here's a bit of information, now evaluate what's in front of you. So they can make us draw a histogram then, not just, yes, they can make you draw a histogram. Going back to this, I feel like the sampling techniques you were given, because obviously you were given that in paper 2, so I don't feel like they're going to go down that route, because that, I feel like they've ticked that one off with paper 2's question. I do feel like rates of enzymes, it's really interesting. The rate of enzyme controlled reaction could be, and this is why it's fascinating. So as I was going through this, you'll be like, oh, but paper 1 had loads of enzymes on it. Absolutely it did. So you've got photosynthesis, you've got respiration, you've got manipulating genomes down here. So if they were to ask a question in this paper and use enzymes, I do feel like that immobilized enzymes and biotechnology would come in this part here. However, moving forward, colorimetry hasn't come up. So I feel that colorimetry could come up in some form of question. I don't feel like pedometer is coming up because pedometer came up last year. So I don't feel like they're going to do it again. Oh, really? Am I glitching for everyone? Is this glitching for everyone right now? Oh, okay. I'm glad it's not glitching for everyone. Do I think respiration and photosynthesis is coming up? I don't feel like respiration, as in aerobic respiration, is coming up. I do feel that anaerobic respiration potentially is coming up. Good. Thank you, guys. Right. So looking at the colorimetry side of things, I do feel like that can come in now. Serial dilutions. Oh my word. Let's try and do this. Serial dilutions could come in with colorimetry. So remember, if you're trying to make some known concentrations, that's therefore going to mean that you would do a serial dilution for those known concentrations. So it's in making sure you know how to do serial dilution. So someone asked me about examples of some questions. So I grabbed one second quickly before starting this session. I'll come back to PAG 6 in a minute. So if you were given a question like this would be a way in which they are assessing, do you actually know how to do a serial dilution? So in this case, if they give you a question with regards to, should investigate osmosis in this case, they could do osmosis, they could do it on glucose, they could do it on a sucrose, they could do it on a what do I want to say here? A protein concentration, if they were doing biuret. They could do it on any of the qualitative testing, and I will show you that in a minute as well. But one thing to note, because someone has asked, how would you improve your accuracy? It's important you know how to do it. So the student was given a 100 centimeter cube of a solution, which was 1 mole per decimeter cubed of sucrose, and asked to prepare 30 centimeters cubed of each. So it's really important you know that final volume. So that final volume is really important because that is what you're going to be using to figure out how much. So if I'm starting with 1 mole, I therefore am going to put in 30 centimeters cubed, and I'm going to put in 0 distilled water. Now, please, 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 please do not make the mistake I just did in my answer there. This is the type of thing where they're assessing you about skills. If they have given you in your table of results to 1 decimal point, you must follow that rule. 0.0, 30.0, you will get marked down if you do not follow the decimal points. It's those little bits that are going to make a difference. I do feel like there's going to be a lot of calculations coming in this exam, and the reason being is because I've not seen that many come through from those 2 papers. So it's important that you do look at what they've given you, and then you're looking at, right, well, how do I get that 0.8 of 30? So if I'm going to work this calculation out, this is where I was about to find my phone there, but I can't use my phone because I'm using it on here. Let's get the calculator up. So I'm not used to using a make sure, this is the other thing, make sure you practice using your calculator. Make sure you don't use a different calculator in the exam. I'm so used to using my phone. I'm now like, a couple of seconds have gone because I'm like, what do I do? So make sure you practice using the calculator before going into that exam tomorrow. It's worse than A-level maths. I agree with you because you really have to figure it out. And I've got a question I want to show you as well on conversion. And this is the thing they do. I completely agree. The paper is either going to be really nice or it's going to be horrible. So you want to take your 30 and you are going to want to get 0.8. So what I'm going to do is I've got 0.8. I'm then going to times that by my 30. So in this case, because I now want to get 0.8, I'm going to times my 30 by 0.8. And what am I going to get? I'm going to get 24.0 that I need to put in of my water, of my stock solution. Therefore, I need to go up to 6 to get that up. So if they give it like this, you're literally just going to times it by the concentration that you want. It's quite a nice one. So 0.6 times the 30. And what you're going to do is 0.6 times my 30. And that therefore tells me that I'm going to be putting in 18.0 of my stock solution. So each time, this is what I'm doing. And then I need to obviously make it up to the 30. So I'm going to put in my 12. Well, it's quite easy because now I just drop the opposite way. 18, and then I'm going to put 6, and then I'm going to put 24, and then I'm going to put 0 and 30.0. So that's quite a nice question if they ask you a question like that. And remember, they will have low level, they will have medium, they will have high level questions. So it depends what way they want to ask that question. So zero dilutions can come in at any point, and it can link to any bit that you are looking for with regards to your marking points. So do make sure, which chat am I reading? On my YouTube channel, Biology with Christine, because I'm displaying the stuff there. With the PAG 6, chromatography or electrophoresis, they've already asked you about chromatography. So therefore, they won't ask it again. If they are going to ask it, it'll be electrophoresis. So therefore, they haven't done the manipulating genomes part of it. They could bring that in paper 3. So just be aware that could come in. So if they are going to talk about electrophoresis, what you have to remember is that your wells need to be down where, I was about to write plus there, but that's the wrong thing, down where the negative electrode goes and the positive electrode is the opposite way around, because your DNA fragments are going to move because they will be repelled because DNA is negatively charged. They haven't asked that much on nucleic acid. So I feel like that could potentially come in and will link as well to genetic engineering. Tony, go and double check my one from yesterday because I did stuff on genetic engineering in yesterday's live if you weren't part of it. Microbial techniques now, although they asked you a question about aseptic technique, I do feel like they could ask you more because I feel like this is where your fermentation comes in. So the importance of manipulating the growing conditions in batch and continuous fermentations could be where they bring in that microbial technique. So I do feel like that will potentially come through in this one because they'll also be able to look at growth of microorganisms. So a practical investigation into a factor affecting the growth of microorganisms. So this could be where they ask you to culture yeast and what factors are you going to be changing, and therefore what are you going to be observing. So your independent variable, what is that? Your dependent variable, what is that? And then how are you going to be able to see that growth that's going to come up? So it's important as you're going through these practicals, these PAGs, that you're thinking about where might they assess me and where haven't they done that already. Guys, I will come back to questions in a minute. Yes, I have had access to the paper too now. I looked at it just before I did this quick section for you. I have seen it. What factors would you change? Well, what nutrients are you supplying the microorganism with? So nutrients would be one of the things that I'd be wanting to talk about. They're going to need, are you going to do it as aerobic or anaerobic? Oh, my life has been restricted because of alcohol, tobacco, and drugs. That's weird. There we go. Supposedly me teaching you about biology is having an effect on the TikTok. That's hilarious. Anyway, back to this. So what am I changing? What do the microorganisms need? Well, the nutrients, the aerobic, or am I doing anaerobic? I have a feeling they're going to go down anaerobic respiration more than aerobic respiration. The temperature. What happens to temperature? So remember, when you look at your fermenter, and we have got a, for example, a cooling jacket. Why do we need a cooling jacket? How do they know that? What does temperature do to the rate of the growth? So if I'm going to change the temperature, what's that going to do? So my independent variable would be that I'm changing the temperature. I would then give them 5 values, whatever those five values may be. So I may start down at 15, and then am I going to go up to 15, 25, 35, 45, 55, 65, 75? How far am I going to go? At least 5 values. And you want to be thinking about the distance, so the intervals between. Don't go too high a jump, because that will have a negative effect. If I'm going to be growing these microorganisms, and I'm going to show you a question on this, by the way, in a minute once I've gone through it. If I'm going to be growing microorganisms, or I'm going to be looking at animals, and I'm doing it aerobic or anaerobic, how am I making it anaerobic? If I'm using animals, I'm not going to want to make it anaerobic, because that would actually be quite dangerous for them. But do I care about microorganisms and having them anaerobic? Is that something to consider? So this is a way in which you need to think about how you can link what you know to the situation. PAG8, that transport in and out of cells, I really feel like biological membranes is coming in. I've said that to you in my list, and this is where it could come in. It could either be on biological membranes and the structure, or it could be related to movement of substances in and out. Now, that movement of substances in and out is linked to so many different places. It's not just about osmosis. It's not just about the beetroot and the colors coming out. It could be, like I showed you yesterday at the end of my session, it could be where you're using a model to mimic translocation. For example, you're looking at that movement of substances one way or another. I think everyone is stressed, by the way, guys. You're not the only ones who are stressed, but I do really feel biological membranes is coming through quite high. PAG9, again, they haven't done anything on PAG9, and I feel like if they're going to do it, they may do it on the emulsions test, which is what I've already said, but they could do it on all of them and look at you being able to see how you would do the testing for it. Now, note, nucleotides and nucleic acids are also linked into PAG9. Qualitative testing, because I'm going to use the emulsion test to actually pull out my DNA for that precipitate. Also excretion, because it can be used for medical diagnosis. So therefore, you need to be looking at all the different ways that they could ask these questions. So when they say it's synoptic and there's looking at those links that are going to be happening, it depends where they decide to go. This is the hardest paper to predict for, because they can go in any which direction. But based on what I've seen from paper 1, paper 2, these are the sorts of things that are going to come through. And this PAG10 is a fascinating one. So PAG10 is about investigations using data loggers. So we talked about that yesterday very briefly. Either using data loggers, so using data loggers, or computer modeling. So it could be where they give you a picture. Now, you were asked about conjugated proteins already in paper 1, so I don't feel like they're going to do that. But again, nucleotides and nucleic acids, I feel like that's coming through, and it's going to link to that manipulating genomes and genetic engineering side of things, maybe. or electrophoresis, potentially. But there's so many ways in which they can look at this one. So it just depends where they want to go. But I wanted to note respiration, photosynthesis, manipulating genomes, they can come again and again. Yes, the ethanol does cause the DNA to precipitate. That is correct. And then the PAG 11, the last PAG that I want to talk about is PAG 11, animal responses. Oh, look at this again. Photosynthesis, respiration, animal responses. Now, this is an interesting one because this is also where they could bring in your communicable diseases, communication and homeostasis, and transport in plants again could come in here. Now, I'm not sure where they're going to go, but my gut feeling is it's going to be practical investigations, respiration rates on anaerobic conditions. I feel like that is where they may go, but I could be wrong. So I wanted to show you some types of questions that could be used and where they could link this together. Do you think much on kidney and liver coming up? I don't know. I've kind of ummed and ahed and gone back and forth with regards to that one. I don't know. I feel like the liver and kidneys came up so much in the last 2 years. And because there's been nothing on muscle, nothing on neuronal communication, I feel like that's coming huge. So I don't know, to be honest, but that's where my gut would say. I don't feel like they're going to do mitosis and meiosis because they've already done that for paper 2. So I feel like they've kind of covered that. Histology could, but last year they did pancreas and they did the liver. So will they do that again? I don't know. I feel like, again, it really just depends what they're going to do. And I know this is frustrating, guys, because I'm not giving you any straight answers because I don't know. I've got my gut instincts of what I am looking at, my research, looking at what I feel. And I even sat today again with my book and I went through. So this is my list now of all the topic areas that I feel were in paper 2. So cell structure was heavy, evolution, patterns of inheritance, ecosystems, mutations, communicable diseases, a lot of module 4, population sustainability, sampling, animal cloning, mitosis, meiosis, stem cells, and microscopes. So I've gone through that. And those are the things that I've remembered and I feel like are huge. So I don't feel like they're going to come up again in the next paper because they've already covered those topic areas. So my predictions for those who weren't here, my predictions that I put out, and I've put out on my videos for the last couple of days, these predictions, I'm not changing my mind. Now, having gone through, I'm still not changing my mind on this. I still feel it's going to be quite heavy on this, but the difference is I will know where they will actually bring it in. The only thing I would add in is I feel like DNA, I didn't write it, but I kind of knew that it was going to come up, nucleic acids, because of my genetic modification. That comes through anyway. You have to understand DNA, you have to understand mRNA, you have to understand protein synthesis to be able to understand manipulating genomes. They haven't done protein synthesis. So therefore, I feel like with protein synthesis, you can link protein synthesis to manipulating genomes as well as biotechnology and fermenters, because that protein that's going to be produced could be the desired substance that we want to harvest from the fermenter. So there's such a nice way in which they link together. Or protein synthesis can come into the nitrogen cycle. I feel like it's coming through. I don't know where, but it's definitely coming through somewhere there. Okay, let's look at some questions then about respirometers. And yes, they could ask you to draw the muscles. If they're going to show you a picture, it could be that they ask you to draw, for example, the dark regions with the lines and the light regions, but I don't know. Exactly is short, just to keep that going. What histology do you need to know? Was that not in my list? That should have been in my list on, maybe it wasn't actually. The histology is the pancreas, the kidneys. I swear it was on my list somewhere. Yes, okay, it's right back at PAG 1. Hold on, let me bring PAG 1 back down and then I'll go over this question here. So I'm going to come down here. PAG 1 is where you've got histology of the nephron, internal and external of the kidneys. You've got the structure and function of the mammalian kidneys. You've got examination, stain section, so histology of the liver. You've got histology of the pancreas. You have also got examination of stain sections or photomicrographs of skeletal muscles. So this is where I feel like that potentially that PAG 1 is coming through. You are going to get some form of histology and it will be linked to this, would be my guessing, because they've done so much on the microscopes already on the stem cells that I feel like it's this one that's coming up. Right, let's actually look at some questions then. Do I feel like the brain would come up? I don't know, because it's a difficult one. I feel like the question that was talking about mitosis and apoptosis kind of linked to the brain and the structure of the developments that would happen. So I don't know. Okay, I wanted to cover this question because there is a nice question that I found that links to this that actually talks about how you would switch this kind of respirometer around if you wanted to look at yeast and the culture. So this is why I want to look at this. So it depends. If you haven't done enough past papers, then past papers are great for practice. But if you've done loads, then no. Just go over the content, get secure on your content for module 2 specifically. I would say module 2. Cerebrum is the conscious thought. Cerebellum is to do with your coordination. So the cerebrum is up here. This is where I've got all my conscious thought that's going on. I am aware of what's going on. The cerebellum is to do with that coordination, and therefore it is involuntary. I'm not thinking about it. Right, okay. This is something I will answer actually, guys. With regards to your mind going blank, in the exam, I know exactly what you mean. You sit and you look at a question and you stare at it, and it's like you're trying to pull the information forward and it's like a blank screen. Someone switched that TV off, okay? That is a problem for you. Now, if you were in the car or you were cycling and there was a roadblock and they shut that road and you could not get to where you needed to go by going a direct route, which is looking at the screen, the direct route, you cannot go there. What are you going to do? You're going to divert the pathway to get to wherever you need to go. You need to do that in your brain. Now, I do that through blurting. So if I've got a question and I am looking at this and I have no idea how to answer the question, I start blurting. I mean, most of the time, I'll actually cover up the questions and I won't look at them. And the reason being is because if I am looking at this question, I start to freak myself out. So you'll notice if you've joined me in any of my sessions, I slowly work my way through and I start making notes to myself. Right, they've told me this is aerobic. So I start making notes and say it's O2. Right, I've got 2 species. I've got different species of insects. So therefore, I need to think about what am I going to control? If I'm not controlling the type of insect, I therefore need to control the mass of the insects that I'm using, because if I've got different masses, that will have an effect on my dependent variable. So what I'm starting to do is I'm starting to give myself trigger words. I've not answered or looked at the question yet. I'm just helping myself know what's going to happen. Right, colored liquid, what have I got? It's oxygen that's going in. So I start drawing. And then I've got soda lime. Well, I know the soda lime is going to absorb the CO2. So therefore the CO2 is going to come down here. And that's therefore going to make this bubble colored liquid move along. Okay, why do I need the syringe? The syringe, this is going to have air in it, and that air is going to be pushed back down here, and that's therefore going to push my air bubble back for calibration and also for repeats. Because I want to repeat my investigation because I want to be sure that I know how to do this investigation to get a valid set of results, because I always want to do repeats so I can calculate my mean and I can also work out my standard deviation. And then once I've got my mean and standard deviation, what I'm then going to do is I'm then going to do a statistical. I'm like now panicking. I'm trying to do things so quick. I'm like, my spelling. And guys, I know my spelling is atrocious. I know I trip myself up. So therefore, I would say if you know that, take the time. Statistical tests. So it takes me longer to process things. So I know that. So I have to take the time. If I don't do that, I am going to make a mistake. Now, if I was asked about a statistical test on these two species, straight away, I'm going, ooh, two species. If I calculate a mean, I therefore want to look at my t-test. So already I'm priming myself as to where it might go. So let's just look at what the actual question was. Give us the units, the valid units, which are going to be used for comparison. So then I need to think about, okay, what's my scale up here? So therefore, I'm going to be looking at a volume, ideally. So if I'm looking at a volume, I'm going to be looking at either millimeters cubed or centimeters cubed, depending on what my scale is above. And so therefore, yes, Sarah E, the key thing to add in is that cyclic AMP, remember AMP should be capital A, capital M, capital P. Cyclic AMP is the secondary messenger. That is correct. That does cause, because it connects to protein kinases. So you've missed out your protein kinases section there. Protein kinases are what are going to move the aquaporins. I don't feel like that's coming up, but I do feel like cyclic AMP is coming up and protein kinases are coming up. I have a strong feeling about that. How they link it depends what they're going to give you as the example. So then you're going to need to note that you are either looking at millimeters cubed or centimeters cubed per minute. And remember, I have different species, so I'm going to do it per gram because I'm going to need to look at the mass in case I have different grams. And so therefore, that would be how to answer that question. So then they're asking me, okay, now other factors may affect the results. So this is where they're bringing in, do you know your variables? Do you have a list of variables that you can go to? So if I'm changing the species, I therefore need to control everything else to be exactly the same. So I need to make sure that it's the same volume of air at the start. I need to make sure that I am going to control, for example, the temperature. I need to make sure that I'm going to control all those other factors that could have an effect on that. So if it's pH you're looking at, if it's carbon dioxide concentration, whatever it is that you are going to be looking at, make sure you have your list. So, and the other thing to know is it doesn't just say describe how one name factor could be controlled. So you're going to state it and then describe how you're going to control it. So exactly, I've got someone who said temperature. Amazing. You said temperature. Now what are you going to say? You're going to say to me, you're going to control this because you are talking about using potentially a water bath. You are not going to get the mark for saying water bath. That's not enough. This is where you fall down if you are thinking, I'm going to control the temperature by using a water bath. No, I'm going to control the temperature by using a water bath at a specific temperature. So I'm going to use a thermostatically, thank you, controlled room, or I'm going to be using a water bath, which is going to be set at a specific temperature, and I will monitor it by using a thermometer. Please, please, please make sure you are giving the detail that's going to take your answer to the next level. It's important because that's what's going to make the difference in you getting the marks or not. Okay, then it asks you to explain why the soda lime in the tube must be replaced when the other species of insect is placed into the respirometer. So in this case, what they're wanting to do is they want to assess you on, do you actually know what is happening with the soda lime? So when the soda lime absorbs the carbon dioxide, that is going to change the available soda lime that's going to be able to absorb carbon dioxide. So you must change if you are going to be ensuring that you are using the variables to be controlled. Sorry, I'm reading these questions at the same time and I'm getting a little bit confused here. So my brain's going, stop doing it. So let me just say that again. With the soda lime will absorb the carbon dioxide. If I don't change the soda lime, what's going to happen is it will not be able to absorb the same amount of carbon dioxide as the previous investigation. Therefore, I would expect there to be less movement of that colored liquid. Now, if I'm talking about pH, and I wouldn't be talking about pH in this example, but if I were talking about a yeast solution, for example, then yes, I would want to talk about using a pH buffer that is going to be used to buffer the pH. Okay, I want to show you this question here though. So this came, another type of question, different exam board. It was really funny. I could see, I love it. For those of you who were here last night, when people have been making comments about, oh, you don't need to know about the latent phase. And someone was like, well, she said that in her session. Yes, these exam questions are not from your exam board. What I'm trying to show you is how they could come in with a similar example, okay? So just so you know, this is not from your exam board. This is a similar type of question, however, that they could ask you. And I like it. This is actually a 9 marker, but I've changed it to a 6 marker. You need to replace the soda lime because the soda lime would absorb less CO2. Thank you for writing the answer there. Okay. Here we need Amna. No, where are you, Amna? Amna wrote me an amazing review on my Trustpilot today. It made me so happy and chuffed to see it. So I'm very grateful to Amna. One, for the fact that she comes to these sessions, because she does help you, but she's actually consolidating her own knowledge by helping you guys out as well. It's one of the best ways you can actually learn. So if you are a year 12 that's here, you are going to be guiding yourself by guiding other people. So it is a really good way to do this. Right, let's look at this question. This question was told about fluoride ions were to reduce anaerobic respiration in yeast. So in this case, what they're looking for is discuss how you would modify this bit of apparatus. So this apparatus that you can see in the picture was a picture that was given in the exam. And therefore, what they did is they asked you to modify this. Now, what they want to do is they're wanting you to see about the fluoride ions to find out if fluoride ions have a significant effect on the rate of anaerobic respiration. So this is why I keep saying to you, this question is actually from Edexcel B. Yeah, Edexcel B, because it was one I was doing this morning with my student who's got their exam tomorrow. It's from Edexcel B. And when I saw it from them, I was like, nice, this is a really good question. But then I was adapting it for you guys for OCR. So one of the ways in which you'll be looking at it is always, when they're asking you to modify a piece of apparatus, you've got to think about, right, well, what are you going to use? Well, yeast needs some form of nutrients. So what are you going to provide them with? Well, if you want them to do respiration, you're going to want to give them glucose because they're going to be doing anaerobic respiration. You want glycolysis to then go through the anaerobic pathway. So we know if we're right and we do this, if we give them glucose and they therefore create pyruvate, we know that they are going to, because it's yeast, produce ethanol, and they do that by releasing carbon dioxide, decarboxylation, and then they're going to produce ethanol, and they produce the ethanol by the fact that the reduced NAD is going to come along and drop off the hydrogens and become oxidized so that it can then regenerate to go back to glycolysis. Okay, so we know that that's what's going to happen. They're not going to ask that kind of detail, I don't think. What they are going to expect you to be able to do is say, right, okay, if I need to use my yeast and they need to anaerobically respire, I need to provide them with a glucose solution. So therefore, if I'm going to give them a glucose solution, I need to make sure the concentration is controlled. So I may need to do a bit of zero dilution to get that concentration. I also want to ensure that my volume of the solution is controlled. So therefore, what I would say is make sure that when you're doing this, that you are actually looking at the information that they've given you and what could they be looking at me. And I've got a bit of a mark scheme to show you for this one. So what's your independent variable? Now, next thing, what's your dependent variable? Well, what are we actually measuring? If we're measuring the rate of anaerobic respiration, we're actually going to be measuring carbon dioxide that they produce. So we do not want to have something that is going to absorb the carbon dioxide. So because we don't want the carbon dioxide to be absorbed, we need to ensure that we modify this by not having something that will actually absorb the carbon dioxide, because we want it to go back. Now, you could look at ethanol concentration, but how are you going to measure that ethanol concentration? No, be careful. Fritz, that's not what they're talking about. They've changed the question. They've said it's to do with yeast. They want you to modify this so that you're now doing it with yeast. So what I would say is make sure that what you're looking at is the question that they've actually asked, and can you apply your knowledge to it? So in this case, just to show you, what am I going to do? Well, I'm going to have a control. I'm going to have yeast and a glucose solution without the fluoride ions. And I'm going to do the experimental side of things where I'm going to have yeast and glucose solution and the fluoride ions, sodium fluoride at a known concentration. So this is where what you would be doing is you are changing it up. Now, what am I keeping the same? Volume, concentration of everything else. Temperature, using a water bath, but remember to say that it's stated at that set temperature. How long am I incubating it in the water bath for? What's the volume of the solutions that I'm using? And then how am I measuring the gas that's being produced? So because it's anaerobic, it's producing the carbon dioxide. We're going to look at this increase in the pressure in the sealed tube, because it's anaerobic respiration I want it to be doing. That's another thing. I need to make sure that it's sealed. And maybe I want to have a layer of oil. So that's another thing to add here that I haven't actually put in. Oh, no, let's try that again. I want to add oil. So if I've got my flask and my flask has got my yeast solution, if I've got oil at the top, that's therefore going to ensure that this is anaerobic respiration. Yes, you would absolutely be removing the gauze because you're going to be using a solution instead. So just so you can see, this was a 9 marker from the exam board, and this was what they actually wanted you to say for that, which is not that different to what we'd want you to say at OCR in how would you modify it, what you're going to do with the method, and then how are you going to ensure significance. So this last part, just to be clear, this last part was all to do with the fact that this was a 9 marker. My question wasn't, it was a 6 marker, but in any type of exam, you always want to ensure that you are giving information about, I'm going to do repeats, I want a statistical test, I'm going to look at that rate, important that we've got the rate of the solution. Okay, so I'm now going to jump around a bit. And I want to look at a Graticule because, again, I saw this question. I thought, ooh, they haven't brought in a Graticule. And so, therefore, I'm kind of jumping all over the place, Liv. I'm doing what I think they could ask you based on what I haven't already done sessions on. I still feel like there may be something on that because they didn't bring anything in, but I don't know how they would bring it. I feel like the protein synthesis is coming in, so transcription, translation, so they may throw in a 1-2 marker on that. How would you know if the lag phase was over? If you're looking at population growth curve, the minute you see your exponential growth, so you get that constant rate, so a straight line. Yes, the liquid's going to move because of pressure difference, absolutely. So this one was an interesting one. So again, this was from EdXLB. But I really liked it because it was a different style of question. I thought, ooh, this is interesting. And so what they did is they told you in this question that your stage micrometer, 0.01 millimeters, completely different way of asking this question. They then told them that the diameter of the heart, whichever way you want to look at it, the diameter of the heart was 0.2 millimeters. So you've been told everything you need to know. You don't have to do the measurement. What you have to be able to do is actually note how that links to this. So they wanted you to know the 0.2 millimeters divided by 0.01 millimeters obviously will give us 20 divisions. Then what we want to do is we want to take 20 divisions here on my stage micrometer and then equate that to the eyepiece Graticule. So straight away, and this is why I want to talk about this question, I've had so many students that I've worked with who've then gone, ooh, it's 10. Be very, very careful when you are reading where it starts and where it goes to and where you're measuring from. So my line goes from 1 to 10. Therefore, it is 9 eyepiece units. Do not get caught out by a question like that, because that is one that does tend to throw a lot of students off. Now, this next question that I want to show you, the reason I want to show it is because although they already did a bit about the cardiac cycle, it's not about that. It is about, do you know how to do conversions? Mindful Mum, I agree with you. I think the histology could be on muscles. They've done it once before where they gave different questions and they actually gave a histology of pancreas, the liver and the muscle. They gave all 3 and they were expecting you to be able to label what it was you were actually looking at. So it's not potentially just going to be about muscles. They may give you a histology of cardiac, smooth and skeletal muscle, maybe, or they may actually just give you a whole lot of histology and then want you to be able to analyze. So it could be any of those ones that I told you about before, because they haven't actually done anything on the liver and kidneys yet, that could be their way of going, tick, we did it. And so that would be the question. Yes, absolutely. You're absolutely right on that paper. Okay, so let's look at this one then. And the reason I want to do this one, yes, you should know the characteristics of striated muscles. You should know the difference between the two, the three of them, whether it's branched, whether it's uninucleated, whether it is so multinucleated, uninucleated. Is it branched? Is it not? Is it striped? Is it not? So is it striated or not? I feel like they could give you a electron micrograph of the skeletal muscles so that they can then test your knowledge on whether you know those different parts. Can I show you? Yes, give me a second. Give me 1 second. I will get the insert and show you the question I'm talking about. So this is the other thing to note. They haven't given you an insert yet, have they? So if they haven't given you an insert yet, it doesn't mean they necessarily will. The inserts are all about if you have got colored stuff they want to show you. Nope, it's not that one. Typical when I need it, because I'm looking for it to do it with you guys, I can't find it. Yes, really annoying. I can't find it. It's typical because I'm trying to do it too quickly. I'll come back to that because I want to cover this question and I want to make sure that we actually do cover the parts that are needed. Okay, so looking at this, and I will come back to the question that I'll find it in a minute, but I don't want to waste too many minutes because obviously time-wise, we don't have that much time. Am I doing Succession? No, Jasmine. Guys, if you really want to just look at specific topic areas, go look at my playlist. The whole content is there for you and it's broken down. I don't feel like Succession's coming up. I may be wrong in that one, but I really don't feel like Succession is coming up because they did it so big last year. So I don't feel like it would be a big one. Translocation, I do feel is coming up. So the reason I want to talk about this one is because you've been given some information and none of it's difficult, I don't think. So you've been told 0.2 millimeters. You've been told that you've got your 4/3 pi r cubed, and you should therefore know that that needs to be divided by 2 to give us 0.1. That's not the difficult part. What I want to show you guys is the number of students that actually 0.1 cubed, will then work that out. And when they work that out, what will then happen is they will get to the point of having a number, which is 0.00 41893, for example, and then times that by 200. Great. So we times that by 200, and what do we get? We get 0.837. Why can I not do this? 0.83786. So there we go. So this is showing us millimeters cubed per minute. Now, one of the things that they always do is they will always want you to note your units, and your unit should be decimeter cubed per minute. So you're going to have to do some form of the conversion, okay? So it's important to know how to do the conversion, and this is what I want to show you. So if you're a mathematician, don't worry about this. When I say don't worry about it, ignore me if you're like, we know how to do this. But for those of you who struggle, because I struggle with maths, and I feel like maths is coming up huge in this paper. So therefore, if I'm going from decimeter cubed to centimeter, if I'm going from decimeter to centimeter, and then going to millimeter, what I'm going to do is I'm going to times by 10, times by 10. So therefore, I know that if I'm going decimeter to centimeter to millimeter, I'm going to times by 10, times by 10. What you have to know is this is going from decimeter cubed, let's say, to centimeter cubed to millimeter cubed. Do not get caught out. Times 10 to the power of 3, times 10 to the power of 3. So if I'm going in the opposite direction, if I'm going from millimeters to centimeters, I'm going to divide by 10. I'm going to divide by 10. So if I'm going from millimeters to centimeters, I'm going to divide by 10, millimeters cubed to centimeters cubed. I'm going to divide by 10 to the power of 3. I'm going to divide by 10 to the power of 3. So therefore, I need to divide this number by 10 to the power of 6. So do make sure that you are going to be using the correct number, the number of students who would divide that by 1,000 and get the wrong number. Please make sure you look at, if it's cubed, that you have done it to the power of 3. Hi, Phil. So do make sure that you go over that, because I do feel like that's the sort of thing that they will do with this type of exam question. If they're going to give you it, they're going to give you a conversion. Now, somebody mentioned earlier about how do you know about lag and log phases? And I do feel like they could bring this in, and looking back at population growth curves, because they haven't really done anything on that. And then what I would say is I will then have a quick skim up. There's been so many comments, guys. Thank you so much. I will look through and see if I can come up with some of the topic areas. Mindful moments. Thank you for trying to help by answering some of the questions. I've seen some of your videos as well, so it's really cool that you're here helping out. Okay, so the graph shows the growth of a population of the same microorganism on 2 different media. So therefore, if this was my independent variable, what I'm changing is I'm going to be changing the different media. So it's important that you are aware of what they sometimes will tell you in the information above. Yes, I will do calculations on that one in a minute. Let me just finish on this one. Identify each stage. Well, what we can see here is this is a slow stage. So you've got here, A is different in each of these. So you know that A has to be the lag phase. And because it's the lag phase, oh, hold on. If you're not the same mindful moment, oh, I thought you were someone who was on the videos that's been doing some revision stuff. I get all these videos that come up on my newsfeed because obviously I'm all about OCR A and teachers who are doing videos. So I thought that was you. So if it's not, I apologize. Lag phase, first part, what can we see? So we've got this lag phase, and then what's happening at the lag phase? The lag phase, they are synthesizing those enzymes, synthesizing the different things. Do you need to know how to draw it? No. Label it? Yes. Jokeman, in answer to your question. Thank you. I am going to do bacterial dilutions. I am going to do it. It is coming. It is coming. The log phase is this part here, where you see exponential growth. You're going to see that log phase, and therefore you've got that A is the lag phase, B is the log phase. We've got different lag phases, different log phases, because we have different things that are happening. A, the lag phase is about that acclimatization. B is about the fact that they are now going through that exponential growth, doubling, doubling, doubling. And then we can see that they have reached a different population growth maximum at that stationary phase. Why might there be? I know I do try. I try and keep up with the questions. It's one of those things that I want these sessions to be about me helping you as well. Don't say that, because that's coming up. For mentors, it's definitely coming up. So one of the things to note in this question is this medium is sucrose, which they've been really nice and told you it is a disaccharide. This one's a glucose, it's a monosaccharide. So therefore, you know, because one is a disaccharide, you should really have sucrase, the enzyme for breaking down the sucrose. So if they are obviously not able to break it down or utilize it enough, they're not going to get to a high enough population stationary phase. Is not going to be high as the previous one. So this is one of the questions where it's about the application. It's not just about the lag, the log, the stationary phase, and the death phase. It's about, do you understand if they're at different points, what is it that's causing them to be at different points? Are they able to utilize them in the same way? What if I said, right, I'm going to grow them on agar, but what I'm going to give them as a nutrient is going to be cellulose. And because they're going to be grown on cellulose, they're not actually going to grow. What's happening then? If they're not growing on cellulose, what does that mean? Well, that means they cannot produce the enzyme, which is cellulase. So if they're not producing the enzyme, which is cellulase, that therefore is something that you can suggest. So remember how I said before about the fact that they will bring in protein synthesis somewhere. It just depends where they're going to go. So in this question, no, this question specifically is only asking about you identifying A and B, the lag and the log phase. It's not asking any more. What they're saying is, if the questions continue on, that's when they start to think about, well, what is it that you're actually growing? Okay. Don't want that one, don't want that one. Here we go. People keep asking me about these tenfolds. Why are we bringing up cellulose? The reason I'm bringing up cellulose on that one is if I were to grow my microorganism on an agar plate. So remember, let me just bring this in a second. Biotechnology, culture of microorganisms. Okay, so remember when we grow microorganisms, whether it be bacteria or whether it be fungi, in this case yeast, we're going to grow them because we can get them to grow in large numbers. We're going to culture them, and that can either be in a liquid nutrient broth or it could be in a solid agar medium. So what I was saying is, what if the nutrients that are placed in this solid medium could be the sucrose even, could be glucose, could be maltose, could be cellulose, could be starch. It just depends where we want to go with that nutrient. That could be the nutrient that we're changing. So if we were to grow them in cellulose, if we were to grow them in starch, if we were to grow them in sucrose or glucose, how are they going to be able to use those saccharides? How are they going to use them? Well, they have to hydrolyze down the polymers or the disaccharides to get them into the monomers to then be able to use them. So only if that microorganism actually has the enzymes or produces the enzymes are they then going to be able to do it. Mindful moments, please don't say this is boring. If it is boring, then off you go. So that's what they would be doing if they were asking that question, okay? So just so you are aware on that front. Now, let's look at a question to do with dilutions. Now, you've been told in this question that the students investigating, oh, okay, I'll let you off then. I was about to say, hold on a minute. A practical investigation on the effect of enzyme concentration on the rate of a reaction. So they've been given a 1% enzyme of trypsin, a supply of distilled water, and some test tubes labeled 1 to 4, and they've got 10 centimeter cubed syringes. So therefore, they want you to describe how you would do the tenfold. So in this case, if I have my original stock solution, so this is my stock solution, which is 1% trypsin. I'm then going to take 1 centimeter cubed out of my stock solution, and I'm going to place that into a test tube. And when I put it into the test tube, I'm going to add in 9 centimeters cubed of distilled water. So what I now have is I now have in this, I have 10 centimeters cubed, but it started off with only 1 centimeter cubed of my stock solution. So I have diluted it tenfold. And then I would do that again, and then I would do that again. However, do not forget to say, before I'm going to move it to my next clean test tube, I need to mix it so I've got an even distribution. So mix so you've got an even distribution. Because if I'm going to put in 1 centimeter cubed and 9 centimeter cubed, let's say the 1 centimeter cubed ends up at the top and the 9 centimeter cubed ends at the bottom, then that's going to have an effect that's therefore going to change what I'm taking out. So it's important that you note. So I was really tripping myself up when it came to zero dilutions and it came to doing the calculations. I kept getting it wrong. And so, yes, you could put 9 centimeter cubed in each one and then add in the 1 centimeter cubed. As long as you are adding in a 1 to 9 centimeter cubed, that's what you want to be doing each time. Yes, 1 in a 10 centimeter cubed. Yes. So let's just look at this then. So here's my concentration. Here's my volume. So what have I got? I've got 1 centimeter cubed. Oh my gosh, what am I doing? I've got a 1% concentration, and I am going to have, let's say, 10 centimeters cubed of 1%. That's my stock. So this is my stock. Now I'm going to take 1 centimeter cubed. So if I'm taking 1 centimeter cubed, I'm dividing this by 10. So I've got 1 centimeter cubed, and I'm adding in 9 centimeters cubed. So my volume now is 10 centimeters cubed, and I have divided that by 10. So I divide this by 10. So I've got 0.1 in 10 centimeters cubed. Now again, out of that 10 centimeter cubed, I'm going to take 1 centimeter cubed, and I'm going to add in 9 centimeters cubed. So again, I'm going to end up with 10 centimeters cubed. So to get 1, I divide the 10 by 10. I feel like I'm shouting. Why am I shouting? I hope it's not coming through as if I'm shouting to you guys. I'm not. I'm just getting like, oh. So I'm dividing by 10, and therefore I need to divide this by 10 to give me 0.01. And I'm going to keep going. So in my 10 centimeters cubed, I'm now going to go down. So if I then go to a new slide, so wait. I'm going to bring this over here. Oh, excuse me. So if I then look at this, and this time I'm going to change what I put down. So this time I'm actually going to take away 0.01 centimeters cubed, and I'm going to use that. So if I take 0.01, this time I've divided the 10 by 100 to get to 0.01. Yes, it would leave them with 9 centimeters cubed after when I do the next zero dilution down. It depends how much you're going to use in your sample. Yes, it would. So therefore, what I tend to do, I would actually put more in. So I'm ensuring that I'm leaving myself with the right concentration. But you're always going to use 1 and 9. 1 centimeter cubed. Because if I'm doing a zero dilution, I'm trying to dilute something down. I'm trying to dilute it down, but what I might want to use when I actually use it is I might only want to use 1 centimeter cubed of the trypsin at a concentration of 1%, at a concentration of 0.1%, at a concentration of 0.01%. So although I am doing a tenfold zero dilution, what I'm actually doing is I'm only going to use part of that 10 centimeters cubed that I had originally. Yes, there is still chance of respiration photosynthesis coming back up tomorrow. Why is it divided by 100? Because I only want 0.01 centimeter cubed this time instead of 0.1, instead of 1 centimeter cubed. Actually, it's not by 100, is it? Because I've gone to 0.01. It would be by 1,000. So how do I, no, it is by 100. Hold on, I'm looking for my calculator. See, this is where I'm like, where's my calculator? So if I've got 10 to start with, and I divide that by 100, yeah, it is by 1,000. I put the wrong number in. Thank you. It's by 1,000, not 100. Thank you for picking me up on that because I put 0.01. If I'd gone 0.1, then it would have been divided by 100, but I put 0.01. So it was important to know what I was doing there. That was me being really silly. I had 10 centimeters cubed. I want to put in 0.01. So now I have actually had to divide it by 1,000, which means I need to divide this by 1,000. If I was putting in 0.1, then I'd be divided by 100. Could it be a threefold? It depends what they want. It's about ratio. So if you go back to this one here, hold on. Let me bring it back down. If we go back to this one that we were doing here, it's about ratio, how much of one compared to the other. So it's either going to be a tenfold or it would be about the ratio that you're putting in, one compared to the other. But they'll make that very clear in the exam when they ask that question. So if I divide that, I'm looking for my calculator again, by 0.01 divided by 1,000, that would therefore give me 0.00001. Now, for some of you, you're absolutely right. You can use the 10 to the power of to help you to work this out. That doesn't work for me, okay? That does not work for me. And so therefore, I found a way that works for you. Do what makes the best for you. So I would say the best thing you can do is figure out what works for you and just try and go through that, whatever they do. Exactly. It is much easier for calculations, but it is a case of who knows what they're going to give you in that exam tomorrow. Realistically, what is this? This would be if we're doing a serial dilution to do, let's say, concentration of, I don't know, glucose. It was actually trypsin, but it could have been glucose, it could have been sucrose, it could have been, in this case, it was trypsin, which was the enzyme. It could be whatever they want it to be. No, it's not. It's someone that's obsessed with respiration photosynthesis. It's that it is one of the things is respiration photosynthesis is connected to everything. This is the thing, absolutely, Grace. So I found, as you just spotted, I made a mistake there, but because I was double checking, I was like, hold on, that doesn't look right. That makes sense to me now. So I find it so much easier to do this than to do it the other way. Because one way they could actually do it is, again, looking at, if you've done serial dilution, let's say I've got A, and I talked about this in my last session, B, C, D, and E. They could ask you about which would be the better one to use if I'm going to grow my plates. So A goes into B, B goes into C, C goes into D, D goes into E. So each time I'm doing a tenfold. So each time I am diluting by 10. So I'm doing a tenfold dilution. So I'm going to divide by 10, divide by 10, divide by 10. And then they always throw a curveball and say either 0.1 or 0.01. So do be careful that you look at what they've said, that they're actually removing from. So I'm going to say 0.1 centimeters cubed is going to be removed. So therefore, I'm now divided this by 100. So each time they take out 0.1 centimeter cubed and they plate it 0.1 centimeter cubed, 0.1 centimeter cubed, 0.1 centimeter cubed. Now, what they could say is they may say that you get 4 colonies there, 2, 4, 6, 8, 10, 12 colonies here. So I've got 4 here, I've got 12 there. I can't tell that one. And the whole plate is just. So they may ask you to actually work back to your stock solution and to estimate how many bacterial colonies were in your stock solution to start with. So if they do it like this, then what they're going to ask you is they're going to ask you to do that step backwards. So what I would say for something like that is where that table works really nicely. So it's the most countable colonies, but not too little. If you go too little, then you're going to lose, I know, right? Then you're going to actually estimate too high. And so that becomes a problem. So the fact is, what I'm going to do if I'm working my way backwards is I'm going to times by 100 each time. And then also, depending on where you're going from, times it backwards by the same dilution factor. And then obviously, if you diluted it in the first place, if A was just 10 centimeters cubed of, or whatever it is of the stock solution, then that tells you how much was in the A part. So everybody has been asking me about zero dilution. So I hope that kind of helps on that one. Right, I have seen a lot on plant response. I'm going to answer some questions now, so give me a second. Tannins are no longer on the specification. You're absolutely right. They were removed. They want you to know alkaloids and they want you to know pheromones when it comes to plant defenses. So yeah, you've got your different types of things. So callose is a way of blocking off the plasmodesmata. So that would be a way of them talking about if a plant pathogen were to get in to the Symplast pathway, how would the plant respond? They would therefore start depositing some callose into the plasmodesmata, and then what you have is after they block off the plasmodesmata with callose, they then lignify it, again blocking it off. They'd rather seal the place where the pathogen is down, stopping them from getting any of the access to the rest of the plant. So basically they shut it off. And why is that important? Because they have meristematic cells, so they can just grow more of themselves. Do I think plant responses are going to come up? They did the germination, so I feel like they've kind of done that one. But again, I don't know. You just go up, go up, go up. Do you think it'll? I don't feel like it'll come up that much. If it does come up, it'll be looking at how the plant is obviously going to detect the chemicals from the pathogen and therefore genes turning on. So this is where they could bring in transcription factors, I guess, in the fact that when you have, right, let me bring this in. Give me a second. Let me bring in, because actually it could link to the transcriptional level of gene regulation. Where am I going? Where am I going? Where am I going? Plant defenses. Where are you? Plant defenses, plant defenses, plant defenses. Right, here we go. So if we were talking about plants producing this callose, as someone's already said, so plants will produce callose for a pathogenic infection, but they also can do it if there's environmental temperature changes as well. It's quite an interesting one. So they gave a question about that to do with environmental changes, but if they're going to ask it, they're probably going to link it more to infection. There are receptors that are going to detect signaling molecules. So those signaling molecules are going to be either coming from the pathogen or these signaling molecules could be to do with those pheromones that we were talking about. So these signaling molecules are going to be detected by receptors. So what do you know about that? What is that linking to? Biological membranes. Receptors are in the cell membrane. They are going to be complementary shapes. Those complementary shapes are going to be complementary to the signaling molecule. Therefore, when they bind together, what we have got is a complex which is therefore going to activate a secondary messenger. This secondary messenger will then switch on a gene within the cell. Now remember, we're talking about plants. So we're talking about plants that have nucleuses because we're talking about eukaryotic organisms. So therefore, we're talking about transcription factors here. We're going to be switching on those genes within the cell. Therefore, that is going to increase the production of the callose. Callose is a polysaccharide that is going to be produced. Oh, carbohydrates, where are they produced? By the smooth endoplasmic reticulum. So this is where you've got a different set of things going on. We're going to switch on the genes that are going to help with the joining together of the glycosidic bonds. And that's going to be done in a place where it needs to be done. So this is where they could link in your cell structure knowledge. They could link in your callose, being the fact that you should know it's polysaccharide, poly, many, but this time it's got different structure, and therefore you need a different enzyme to be able to catalyze that reaction. So therefore it's not the same enzymes that are needed. They're only going to produce these enzymes. They're only going to transcribe and then translate these enzymes when they are needed to make callose. Why waste the energy doing transcription translation to block off these plasmodesmata if you don't need to? So again, it's important that we note this will be a signaling molecule that is going to be detected, and therefore we've got that secondary messenger switching on the genes. They are beta 1 to 6 and beta 1 to 3 because we know that we have a different molecule that's being used. Look at the numbers because they actually gave it in the exam, this structure. Carbon 1, therefore telling you that's carbon 1. So you should know if it's 1 there, it goes 1, 2, 3. That's a 1 to 3 glycosidic bond. And then up here, we know that that's carbon 6 because carbon 6 is one that's got the H2OH group. And obviously that's going to block off the cell membrane and the plasmodesmata, the cell wall, and the sieve plate, therefore ensuring that whatever this pathogen is, it's not getting throughout the plant. So that's important for us to note. So that could link to the topic area. They haven't done much on communicable diseases. They've done very little on that. So therefore, yes, they could bring in the other parts. Try to think where they could go on this one. They may bring in oh yeah, and then lignin's going to thicken and strengthen that cell wall. They could bring in the different ones that you need to know. So make sure you do know which ones and how they can actually be transmitted. It's no longer actually going, you don't need to know the difference between whether it's direct transmission or whether it is indirect transmission. But one thing I would say to you is know about spores. If the organism can make spores, that can then be contaminating the soil, or those spores can actually be thrown out, and therefore you've then got the vectors of the wind and the water that is going to transmit these pathogens as well. So it's important for you to note that us as humans are vectors. I saw a question which was talking about, actually it was in your paper too, and it was about how did what was it? How did the predators get there? And it was about humans. Well, we not only bring with us things like we brought the dogs to the Galapagos Islands that fed on the dodo eggs, and therefore the dodos go extinct. What we've also got is we've also got the fact that the things like we go out walking, we walk past plants, and if plants have got a pathogen on it and we walk past them and then we walk past another plant, we can then indirectly pass on that pathogen to another plant. So this is again a way in which they could look at it. Okay, so again, can I just make you very aware, for those of you who haven't been coming to my sessions, I saw a message here that says you could write everything right and still be done dirty by the mark scheme. Zara, actually, no. It's only if you write incorrect science will you get done by the mark scheme, okay? You could write an answer that isn't absolutely exactly what the mark scheme is wanting, but you've described it scientifically correct, and therefore you can get the mark scheme, get the marks, unless it is underlined. That wording is bang right. They want you to put that wording in there. But if you are going to explain it in your own way with enough detail that the science is not incorrect, you're not going to lose the marking point. We would much prefer you to write less and be accurate than to write big waffly stuff. But what you have to realize is that mark scheme that you are using to mark these past papers was the mark scheme that was used for the cohort who sat it that year at that time. Okay? The mark scheme that's sitting there right now for your exam is sitting there with a set answer. However, it will change. After the senior examiners sit down with your papers, they'll take a sample, they will mark it, they will modify that mark scheme for common themes that are coming through to say, yes, we will accept this, or no, we won't accept that because it's not given enough detail. So it's important you know that just because you wouldn't get it in a past mark scheme doesn't mean that the mark scheme that you're getting and the way you write it means you won't get it. This mark scheme is not finalized completely yet. There were last year 20-odd thousand students who sat that exam. You are never going to write in exactly the same way. So therefore, it comes down to the professional judgment of the person who's examining you, but based on extra bits of information that you don't get to see, your teachers don't get to see, only examiners get to see on the exams that they sit and mark at that time. And there's a reason for that, because it's got to be consistent. They've got to do moderation. So if you've done any kind of exam that has coursework, that exam that's got coursework, your teacher will mark it. If it is an internal assessment, they will then send that out, and a moderator will say whether or not they agree or disagree. And your teachers should actually be moderating your mocks that you are doing so that you get consistency within your skills. You can see where those marks will come from. You won't know any of this because you're not in the background seeing what's going on, but there's a lot more going on than just the marking of your papers. And that's why it takes all the way to, what is it, the 20th? No, 20th, 21st of August. It takes all the way to that point because they have to do this rigorous process of making sure your papers are marked in the right way. Now, that doesn't mean that the examiner won't make a mistake. So if you get your paper back and you are very close to a grade boundary, you want to, if your mark comes back and you were like 4 to 5, I've seen it go up a maximum of 9 marks up, but if you're close to a grade boundary, ask your school to download your papers and get them to double check it, just in case there's any miscalculations. I know fine well when I've marked my class's tests, and I've gone through and they've calculated, they've like, Miss, you missed a mark here. And so therefore, that can also happen in your exam. So therefore, what I would say to you is if you're close to a grade boundary, just ask them to download it and double check whether you agree or disagree. Double check whether, ask your teachers whether they agree or disagree, whether they think that you've been harshly marked. I can't guarantee it, but it's always worth you going and looking at whether or not it is going to help you if you are a couple of marks off, okay? I wouldn't say if you're in the middle of a grade boundary, there's no point in going anywhere. If you're at the end of a grade boundary, so if you're at the bottom end, don't get a remark because you may fall into the grade below. So if you are close to the next grade up, then potentially look to get that remarked. But it's not remarked. First, they check and calculate it to make sure they've added up all the points correctly. And then only if there is some discrepancy and they think that is necessary, will a chief examiner remark it. But that's a whole different process. And you have a window in which you can actually have that marked. So do make sure as you get those results back that you do look at where you lie in that grade boundary, and you will be able to see that. Okay. Yes, you do get refunded the cost if your marks change. Absolutely. If your uni lets you in regardless of the grade, then don't worry about getting it remarked. I was praising you earlier, Amna. This is the first time I've actually seen you come in here. I'm missing half these questions. I'm sorry, guys. I was just talking away. Right. If you achieve all the marks that they accept for a question, but then write something incorrect, but not contradicting, would you get marks deducted? Yes. So if you're going to write something that is incorrect and it's not an ignore, if it's incorrect, you will lose marks because it's incorrect science. You need to make sure that you are not writing anything that's incorrect in your answers. And that's why we advise you not to write too much. No, it hasn't come up in paper 1 or paper 2. I feel like nitrogen cycle is coming. Yes, let me go through. Give me 1 second. I did do it in my last one with the session, but let me just see if I can find the, because I pulled the notes across, didn't I? Did I do it in last one on paper 2? Yes, I did. So I talked about histograms. So this was the only histogram that they have actually asked students to draw in the past couple of years. So it's important when you know about the histograms that you are looking at what is your range that you're going through. So when you're drawing along the bottom, your axes, you need to look at what your lowest number is and what your highest number is, but make sure you don't draw your width of your bar too small. So each of these, as I was saying before, was, and I'm not going to draw the actual thing out, but let's say rough, I'm going to sketch it. There's 10, 48, 121, 100 and, oh, hold on. I did that one wrong. Hold on, what have I done? Oh, I started in the wrong place. See, even I did that wrong. That's why I'd say just double check. So there was 10, 48, 121, 130, 100, oh, crumbs, 119. And then this one was 46. Now, if I drew my graph like that, I'm losing marks. Straight away, I'm losing marks. Frequency density is accepted. It would be accepted if you were to do frequency density. That's fine as long as you put along here frequency density rather than frequency. So I was just doing frequency, but you would be able to do frequency density. Your bars must touch and they must be the correct width. That's important. One of the things that I would say is I'm losing marks because the graph space that they've provided me with, there's 50% of the paper and I've barely touched it. So I'm not getting that marking point because I haven't touched that. Yes, you can. HCG in pregnancy, yes, you can, but that came up in not last year's, the year before. No, it was last year's one, wasn't it? Um, I know it's weird, but they will accept it because they're not expecting you to. If they've given it to you in that way, they're absolutely fine with accepting it that way. What's HCG? HCG is the hormone that you produce when you are pregnant, if you are female, and your embryo has implanted into the lining of the uterus. You start producing HCG, which basically keeps your corpus luteum from degrading. You don't need to know about that. But the HCG you do need to know because you need to know about monoclonal antibodies and the fact that HCG is a hormone and you can have an antibody which is specific to it. Yes, it's under the hormonal communications, right? Absolutely. Translocation, I do feel like it's coming up, but I don't feel like it's going to be straightforward. I don't feel like you're going to get a question which is going to be just where's the source, where's the sink. I think it's going to be very much application about what's in front of you. It's also under excretion as well. It is for OCR. Absolutely, it is for OCR. What do you need to know about anabolic steroids? You do need to know about anabolic steroids and the fact that they can be detected in urine. How much will we be writing for 6 marker? You probably lose, depending on how much incorrect, probably lose at least 1 mark if everything else is correct. I would say. What's your language, Matteo? Matteo? Don't know how to say your name, sorry. Oh, the black line in this is me showing that I've not used 50% of my paper. If I haven't used 50% of my paper, I've automatically lost a mark because my scale needs to be big enough that I should fill, whether it's a biological drawing or whether it's a graph, you should fill 50% of the paper that's provided at least. Bars should touch, absolutely. And so therefore I'd lose it because I didn't touch them there. Yep, absolutely. I'd lose marking points. Ooh, that's an interesting one. Could you detect it in an emotion test? I doubt you'd be able to get the concentration on that one. So, Tony, nitrogen gas is fixed to ammonia or ammonium ions. It's not fixed to nitrate. You need the bacteria that have the enzymes to do nitrification. So nitrogen fixing is about the nitrogen gas going to ammonium ions or ammonia, depending on what they're looking at. I have a feeling, LAL, I've got a feeling that dialysis tubing might come up and they may use it as a model, but I don't know whether they'll bring it up as dialysis or not. I don't know. Well, I haven't drawn my scales, so I'd have to double check my scales and I'd have to check my plotting. But I've just done this as a quick sketch. So I want to say it was either 21 or 22. I can't actually remember. No, I don't feel like that will come up about gas chromatography and spectrometry because it came up last year. So I don't feel like they'll bring it up this year. The other thing I would say, Amna, just to add into what you said about bacteria produce desirable products during the stationary phase, they might produce it during the log phase if it's something for active growth. So if we were using yeast and we were wanting to grow beer, oh my gosh, you can tell I'm getting tired. My brain's stopping functioning. If you're wanting to produce beer, you're wanting to ferment it to get the alcohol, ethanol and then ethanol is produced during the active growth phase. And so therefore that would be during the log phase. So be careful that you note that desirable product. Is it something which is a secondary metabolite or is it a primary metabolite? Which one are we wanting? If it's a primary metabolite, it'll be during the log phase. If it is a secondary metabolite, it'll be during the stationary phase, like Amna said. So just be aware of that. They could try and trip you up there because they know students will memorize stationary phase, secondary metabolite, and then they throw the curveball in and ask you about something that's a primary metabolite. So if it's the active growth, make sure you know that that is the log phase, and that's where you see that straight line in the graph. And there you go. That's exactly why what I was just talking about. If it's continuous, absolutely continuous, you'd want the exponential growth, and therefore it would be the log phase. Do we need a title for a yes, you do, absolutely. I don't feel like Hardy Weinberg will come up again. No. Yes, I do feel like muscle contractions coming up. What else? Oh, so the frequency density is just basically, I have 100 between those two. So it would be 10 divided by 100. So the frequency would therefore be 0.1. Then I'd have 48 divided by 100. So the frequency would therefore be and so forth. And then at the bottom one, I'd have the 46 by 200. So the frequency density would be. Histology, feel like something is hard to identify structures. So that with the histology ones, it's an interesting one because I feel like they've already covered differential staining to you, so therefore they wouldn't do that part. So if they're going to go down the histology, like when they did the pancreas, they were looking at, did you know that the islets of Langerhans stained lighter? So if they are going to go down that route, it could be looking at the fact that if you're going to do differential staining, does the cell have lots more nucleus and therefore more DNA? Or does it have more protein? So using differential staining so that you can, so differential staining is where you use it for cell structure to visualize down the microscope. So if you're going to use differential staining, you're going to use that as a way of identifying. So yes, it could come in with identifying the different bacterial colonies if they decide to do that. One thing I would say, guys, is I am going to finish in about 7 minutes because I feel like 2 hours is a long time to be going in my brain. I haven't had dinner either, so I need to eat as well. And today's been a big day. I've been teaching 4 and a bit hours before I then sat your paper 2, before I did this stuff for you and came on here. So I definitely need a break. And I would say to you as well, make sure you have a break. Make sure that you are taking the time to have a break, to have a good night's sleep as well. What about DNA replication? DNA replication could come in because nucleic acid nucleotide hasn't come up. But guys, if you do feel unconfident and want to look at content, all my videos are there. They're short videos. They're not long videos on very specific parts of the topic area. Link to a playlist, which is for your specification point. If you're doing a QA, that's a completely different exam board. This is OCR I've got. How was paper 2? It was an interesting paper. I found it quite confusing on some of the bits, but I also think I was doing it very rushed. I got access to it at what? Hold on, let me think this through. I got access to it in my last session, so 5 to 3. I then had to go pick up my girls. I came back from picking up my girls. We cycled home. And then I sat in the office and tried to do the paper whilst my youngest, my 5-year-old, kept coming in and being like, Mummy, come do this. Mummy, come do that. So I wasn't able to focus on it in the same way I did for paper 1. But I would definitely say that there were some questions that made me go, ooh, I wonder what they want you to say there. I'm not entirely sure. So I would have had to really think about that question. No, I don't think they're going to ask about HCG. I don't feel, and I could be so wrong on this. I'm worried. This is the thing. I've been nervous since I put my predictions out there because I don't know. I'm making an educated guess based on all my research, based on all the papers that I've done. I may get some of it right, but I may get some of it wrong. So I don't know. What should you do? I think you should take a break, step away, trust that you have done enough and you are doing enough. Give yourself the night to formalize your answers. And in the morning, have a quick look over things again. Don't study for hours, 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 because what's going to happen is your brain will be overloaded and you will actually perform much worse tomorrow than if you take a break tonight. I promise you. I promise you, you need to go to sleep. I promise you, adrenaline will help you tomorrow to find the information. And if you do come up against that blank wall, absolutely no, you blurt on paper every single thing, even if you think is the biggest pile of absolute drivel, write it down because you never know, you might be able to gain some marking points. Oh, thank you. I hope I'm so worried because I would hate people. It's why I will never do predicted papers. I've got my workbooks, I've got my study guides, which are practice papers, but I will never do predict papers because I don't know. I might get it right, I might get it wrong. I knew control was coming. I had a feeling, which was why I therefore did my workbook for cellular control and got that out as fast as I could. And I haven't done the rest because I wasn't able to do much. Oh, thank you. Guys, one thing I would say after your exams are over, because I've only been doing this for a year. Okay. I'm literally 1 year and 3 days in for my YouTube channel. And so I took the plunge last year to do this full time. Amna, I was praising you for my Trustpilot review. Thank you so much. And also, it's kind of the nice things that I get to look back on to remind me of all the people that I've helped. I still have every single thank you card that any student has ever given to me up in my loft. I actually have a picture, my first year of teaching. I have a picture that my students, these were year 11 students, they actually made a picture. It was so funny. And it was of a tortoise, and there was me, and they cut out a picture of my face, and there was me with the lead attached to this tortoise, and 5 students with their little pictures attached to it. I still have that in a frame upstairs in my loft. Honestly, I absolutely love when people are appreciating what I've done. I want to help you guys as much as I can, which is why I've done this. I'm definitely going to look forward to my break before I start this all over again with next year's year 13. So the one thing I would say as well, if you know current year 12s, tell them about my stuff, get them, because honestly, if you guys had found my channel before, you would have been able to do it over and over again. Amna's been like, there's like 5 students I will name: Amna, Tony, Chidima, Alex, and V. They have been my students who have basically come to every single one of my sessions that I've done and asked me questions and pushed me and done what. And and I would say that it's one of those things that we do this because we care. And also, I wish I had something like this when I was your age, because I wish that I had some way of turning to someone who knew what they were talking about that could help me. I do a lot with research students, actually. A lot of my private clients are research students. So absolutely, yes, you could see me again if you need to retake. Because actually sometimes a retake is all it takes, taking 1 year out. Oh, oh, is it not? What's wrong with it? That's good to know. What's wrong with it? What have you done? Have you crashed it? What's happened with it? Let me quickly log on. It's still a little bit time, but I do want you guys to have a sleep. Um, who just said that about AIQ chat? I've lost you. Where are you? Ah, where are you? Where are you? No, I don't feel like 2025 is harder than 2024. I think they're about on the same par, and I think your grade boundaries are going to be the same. Guys, please stop being rude. Please stop being rude to each other. That's not okay. I don't like that. So don't do that in my sessions. It doesn't seem frozen on. Oh, I'm so pleased. Seriously, I'm gonna finish now because this is getting to the point where I don't like people who join in and start taking the mic. Everybody is going to have their own different stresses and everyone's gonna have different strengths and weaknesses. So I would suggest what you do now, shut off social media. Obviously now I'm finished. But shut off social media, get down into your own space, have a good night's sleep, have a good breakfast, have a quick look over your own notes tomorrow before going in, and that's it. No more. Don't do anything else. And then obviously tell me how it went and what came up because I'll be dying. I'm literally gonna sit here not knowing what to do with myself tomorrow because I'm gonna be like, come on, come on, come on. So yeah, guys, have a good night. Thank you all for joining me. I actually got up to 300 people watching me on YouTube tonight. That's crazy. That's like super crazy. Um, so thank you, and thank you for sharing, guys. And yes, good luck. Seriously, good luck. Take your time, read the question. You will just try your best, okay? Right. Goodbye. Enjoy your evening, everyone.